Hello everyone, I'm Dr. M. Thirumurthy, Associate Professor, Department of Genetic Engineering, SRMIST. Today, we are going to learn about what is the criteria to select the best PDB structure before going for the structural bioinformatics work. Okay, so where we can get the protein three-dimensional structures. So we can get the protein three-dimensional structures using X-ray crystallography, NMR, and electron microscopy. In the absence of experimental method, we can go for computational methods such as homology modeling or threading methods. So we'll have a basic introduction about how this X-ray crystallography method works. So we get isolated protein, interested protein solution, then this protein needs to grow into a large crystals in which their atom positions are fixed in a repeated ordered fashion. So these protein crystals, right? So these protein crystals are then mount. So when we pass this crystal applied to the X-ray diffraction beam, so we can get diffraction patterns. So because this X-ray crystal structures basically they consist of electron clouds surrounding the atoms in crystals, producing a regular pattern of diffraction. So these diffraction patterns can be converted into electron density map here. So once we get the electron diffraction patterns, then we can use Fourier transform method that can be converted this diffraction pattern into electron density map, and then we can get the protein three-dimensional structures. So this is very tedious process. Okay, so look at the diffraction pattern for this um, um, pattern for benzene and 5-member ring molecules. Imagine that if we have a protein chains which consists of hundreds of amino acid, then imagine the complexity of to solve the X-ray diffraction pattern. Even a small molecule, this will show this much of many different diffraction patterns, like tiny dots there we are getting it. From this we have to convert the electron density map from them. From there we can convert into the protein 3D structure, which is very tedious job. Okay, so once if we go to any kind of PDB structure, basically we can download the PDB structure into PDB format. Okay, so this is the typical PDB format, basically which consists of header, title, atom, and head atom. So we'll have a detailed discussion about what is that header describe, what is the title, what kind of informations we can get it from the atom, and what kind of informations we can get it from the head atom. Okay, so what is the use of header? Header basically provides the overview of the protein and the quality of a structure. So basically, which contains information about the name of the protein, or protein, or the sources of the organism, bibliographic reference, methods of structure determination, resolution, crystallography parameters. We can get the protein sequence. If there are any kind of small molecule information presence, we can get all the information from the header. Okay, so after that, we have other important column called as atom. So basically, the atom represents proteins or nucleic acid information, nucleic acid components, or we can say that. Head atom basically represents small molecules, cofactors, or water molecules present in the protein structure. They'll be represented in head atom. Okay, so after this atom column, we can see the column. You can see the column number, right? 21, 22, 23. So this describe the number for the particular atom. So this particular atom number 21 represent the histidine residues, Histidine residues, they have the atom, nitrogen atom, right? The particular nitrogen atom, they have the number 21, right? So this column represents the amino acid or residue number or residue name. Residue name, which this column represents the amino acid name. And after that, we can see the symbol A or B. So this represents the whether this particular PDB structure they belong to A chain or B chain. After that, we can see the number again. This number corresponds to the amino acid number 3, 4, right? So you can see every amino acid they have like a, so when you see the structure of amino acid. So what is the structure of amino acid? Basically, the central carbon atom attached with the amino group, carboxylic acid, and then hydrogen atom. And then various we have side chain substitution, right? So each and every amino acid they have like a carbon atoms, nitrogen atoms, right? Oxygen atom. So we can get the individual atom position for every amino acid, right? So after that we can see three different columns, right? 55.365, 27.866, 62.971. So this column represents the XYZ coordinates. So this real number represents the atom position for the individual atom, right? So because of this XYZ coordinates, we can get the atom position for the individual amino acid, individual atoms amino acid, right? Okay. After that, we can see very important column called 1.000. This column name called as occupancy of atom. So when we select any kind of PDB structure, we have to make sure that this column, all the atom position confirmation should be 1.00. If it is 0 confirmation, then we have to check the confirmation for the particular atom. But by default, most of the atom, they have the confirmation of 1.00. So after that, we have other important column called as B factor. So what is a B factor? Basically, the B factor represents the temperature factor, so which indicates the static and dynamic disorder in the crystal. Okay, and finally we can see the atom types. Look at the other example. Here you can see this particular, what is the amino acid here? Here we have amino acid called valine. This particular valine, right? They have the atom, nitrogen atom. So this particular nitrogen atom have two different conformation, 0.55 and 0.45. So what is our rule? The occupancy of atom should be always 1.0. Why? How they get two different occupancy of atom for this particular atom? Because the first confirmation has given the occupancy of 0.55 and alternate confirmation 0.45. This particular atom has two confirmation. But only few programs that can process and delete alternate confirmations completely. That's what I said. We have to make sure that when we select any kind of PDB structure, we have to make sure that all the atoms they have the occupancy of occupancy of value should be 1.00, right? Fine. Okay, finally we'll see what is the best criteria to choose the better crystal structure. Apart from the PDB format understanding, so very important criteria in PDB structure selection is resolution, right? So what is a resolution? Here we can see for example, suppose if we take any kind of protein name called JAK2 protein. So the JAK2 protein, many structure has been solved. Suppose if we take three different or four different types of JAK2 protein, which has a resolution of 1.0 Armstrong, 2.0 Armstrong, 2.7 Armstrong, 3.0 Armstrong, which is solved by the X-ray crystallography, all the four methods. Then which structure we have to select? We have to always select 1.0 Armstrong. So which means that the resolution value number should be very, very less. Lower the number, here you can see The lower the number, which means that better resolution. See the resolution of 1.0 Armstrong. Clearly we can see the atom position for the entire protein. So that's what the lower number that will give best or a better resolution. Look at the same higher number, 3.0 Armstrong. Higher the number, that will give lower resolution. So we can see some kind of amino acid will be there, but surely we cannot differentiate what kind of atom they have it, what kind of residues they have it. So that's what, when we go for the structural related studies, we have to select quality PDB structure. I hope you have enjoyed this lecture. Thank you.